Now we have studied about DNA, we have studied about proteins, we have studied about codons, we have studied about anticodons, and onwards to the last part of all of those combinations, we are going to be talking about gene mutations. Without wasting time, without any further ado, let's just assign a formal definition to a gene mutation. Now, a gene mutation is the altercation or changing of the sequence of base pairs in a DNA molecule. Now, DNA, we know it has a specific preset sequence and order of nucleotide bases. But those bases can sometimes be altered. If those bases are altered, what happens? Altered bases, altered mRNA transcript, altered amino acid sequence, altered polypeptide, altered protein. So, an DNA molecule resulting in the formation of an altered polypeptide. We don't get what we want, and this is honestly the basis of so many human diseases. A genetic mutation, because the desired protein is not produced. We're producing a different polypeptide. We're producing a different protein. So what happens? Altered cellular function. That is what we call a diseased or human diseased state. Now remember how we talked about the genetic code and its degeneracy. What did we mean by degeneracy? Multiple, where's that? Nope, right here. Multiple codons can code for the same amino acid, and you will see how this degeneracy helps a lot when it comes to mutations and safeguarding our genome. Now, the degeneracy of the genetic code. The genetic code ensures that most mutations do not alter the polypeptide. Okay, even if mutations occur, sometimes it might not really alter the polypeptide, or only slightly alter it, not enough to cause some serious damage. Not enough mutation, not enough mutagenic factors to cause any serious damage. Now let's look at the types of mutations. You have three main types of mutations, or more like types of, let's call it three main types of mutation, or you can think of it like three main ways in which mutations can occur. The first kind, I'll use a different bold color. Not a bold color, but like a shiny color. Can I get a shiny pink? No, I'll get a shiny blue then. Okay, I'll get a very shiny blue. The first kind is the insertion of nucleotides. Don't try to understand what each of these things are. I will provide small examples and that's going to help you. Then, if insertion is a part, deletion is also a part. So, deletion of nucleotides. And then the third kind is going to be the substitution of nucleotides. Three main ways, three types you can call it. So let's start with the insertion and see what's actually going on. Insertion of nucleotides. I'll just call it insertion to preserve our time. We don't have that much time. Okay. I'm going to type this out. So this is what happens. Simply a, whatever it is, nucleotide with a new base is randomly inserted. Let's expand that. Inserted onto the chain. Now the amino acid synthesized is changed along the entire frame. And we call this a frame shift mutation. I'll take that down. We call this a frame shift mutation. Okay, highlight that word. Let's first understand what we mean by a frame shift mutation. This is what happens in the insertion mutagenic range. Okay, I have a polypeptide chain. I don't know why I keep drawing not the polypeptide chain, the base chain. I don't know why I keep drawing the base chain like this. I can just draw it straight. Okay, let's imagine I have these specific bases in this order. A, G, U, U, C, A, G, C. Right? This guy is going to code for this. This codon will code for this amino acid, and this codon will code for this amino acid, like so. Right? Now let's say an insertion happens, meaning a completely different nucleotide just gets inserted somewhere here. Let's say here. Okay, so this is what is going to happen. It gets inserted here. So because of that, all these things are going to have to move a notch down like this. Okay, now a new guy is coming. He is being inserted here. I'll just reduce his size to match the others. Let's give him a proper mounting point as well. Right. Now, if this transcript is being read from this end, the first amino acid is fine. Still, nothing has changed. Nothing has changed. We still have the purple polypeptide. But do you see? Now, when we're reading this transcript, the next three codons become these three. Not these three. Previously, these three were what we wanted and they coded for the specific amino acid we wanted. But it's no longer that. It's this. And tell you the thing that doesn't code for the amino acid we need. It's a completely different amino acid. And then again, do you see? Again, a frame shift has occurred. We're reading this now, so it's not going to produce the orange one we need. It might be producing this green one. Do you see? We wanted one polypeptide chain with three specific amino acids. Now we have a different one. The first one is the same, but the second and third ones are now different. Why? A frame shift mutation has occurred. Because of the frame shift mutation, everything is being read in a different manner. So the entire frame is different, and this is a huge problem. We usually say that this has a knock-on effect. Knock-on effect. This has a knock-on effect. Okay, if you understand insertions, you can clearly understand deletions. It's the same exact principle. One thing gets deleted. Number 2, deletion. Let's type things out. When it comes to deletions, now a nucleotide with a new base is randomly, no, not with a new base, a nucleotide with a base is randomly deleted. All of that, boom, gone, left. So if I consider the same exact base segment, so let's say we're reading again from the left. So 1, 2, 3, 1, 2, 3, 1, 2, 3. Let's say we are reading that. This guy gets deleted all of that. So again, that's gone. So they have to jump back. to fill that space like this. To fill that space like this, and then again our reading is going to change. A different codon is being read, and then a different amino acid is going to be inserted, different polypeptide chain, different protein. As randomly deleted changes amino acid, amino acid synthesized, and we have a frame shift mutation again. We have a frame shift mutation with another knock-on effect. Insertions and deletions are very similar. Now the third part, the third kind is going to be the most important kind, or I can say the most obvious kind. These are substitutions. These are substitutions. So we're substituting nucleotides. Let's write down what happens in substitutions. Now a nucleotide and its base is randomly substituted or swapped. That's a better way of explaining substitution. So it's randomly swapped for another. All of a sudden, we're swapping a random nucleotide. Now, here's the interesting part of this. Just one thing is swapped, so we're not inserting anything, we're not deleting anything. So there are no frame shift mutations. I'll explain this using a small diagram in a bit. This will only changes that. This will only change that particular amino acid. No frame shift occurs, and there's no knock-on effect on the entire growing polypeptide chain. And these can come in three specific kinds. Substitutions can occur in three specific ways. The first specific way is known as a silent mutation. And these are the best kind. We love these kinds. The first is known as a silent mutation. Then we have something known as a missense mutation. And then finally we have something called a nonsense mutation. What nonsense is this, right? What am I talking about? You'll see in a moment. Okay, silent mutation looks something like this. In a silent mutation, you will have no altercation of the amino acid. Why? The reason being the genetic code is degenerate. Okay, let's look at this in this manner. We have to go back to our genetic code diagram. Okay. Now let's say something like this happens. I have this specific codon to be read. If you refer to the table / diagram, you'll see if I have CCU, I'm going to code for proline. So let's give proline this color. Right, this is proline. Now in a substitution, what's going to happen is one nucleotide is going to be substituted. Right, so something like this. This guy is gone, he's dead. He's being replaced by another C. But do you see, there's no change to the amino acid. It's still this colored amino acid. Why? Because do you see, CCU and CCC all code for proline. That's the degeneracy. That's the degenerate effect of the genetic code coming into play to save the day. Do you see? No alteration of the amino acid. Polypeptide chain is not altered. Protein synthesis is not altered. That's the beauty of silent mutations. There's no alteration of the amino acid. Why? Again, the genetic code is degenerate. Now, what about missense? Here's the thing with missense, very simple. Missense alters a single amino acid. Missense alters a single amino acid. Just one, okay? Just one. This is the worst kind that you can possibly have. Whenever you have a nonsense mutation, a premature stop codon is created. A premature stop codon is created. Okay. And then what happens if you stop protein synthesis all of a sudden? Therefore, the translated polypeptide translated is incomplete. So you only get half of whatever you get. If you're making an enzyme, you get half the enzyme. It can't carry out its functions. You're basically going to die under that metabolic process. Incomplete and lacks. I'm so done. That's why my handwriting is giving up already. That's the first thing usually to go when I am so tired, my handwriting. And lacks shape and function. Right? It's like this. Say you were producing this polypeptide. All of a sudden in the base pairs, a premature stop codon was created due to a nonsense mutation. And this stops. Instead of creating this stops somewhere here. And you only have this part now to work with. It's not going to fold into the proper protein and it's not going to carry out its important metabolic function in any manner. Therefore, we have a problem. Just a small recap. DNA mutations can occur in three major ways: insertion, deletion, or substitution. Insertions and deletions both produce frame shift mutations. They're bad. That's all I'm trying to say. They're bad. Substitutions are better. Okay, substitutions are better because you still have a chance of it being silent, nothing is being altered, just one amino acid being altered. Maybe that's not going to have that much of an effect of it, but the worst kind of substitution based mutation would be the nonsense mutation type, where you get a premature stop codon and the protein or polypeptide chain produced is going to lack function, shape, and overall metabolic function.